My name is Noah. I'm a proud SSP 2024 alum, Purdue Genomics. I'm also an incoming freshman to Stanford University, and I'm hoping to major in computational biology. Um, I work in the Natterson-Horowitz Medical Phylogenomics Lab. I've been working there for the past 3 years or so, and we've published 12 publications together. This specific presentation that I'm giving now is from a 2023 research project that I presented at the International Society for Evolution, Medicine, and Public Health. But more recently, we've been working on more computational bio stuff and kind of using and leveraging bioinformatics softwares to target and understand the evolutionary origins of genetically heritable diseases. Just kind of before we get started, this is more of a shout out to SSP that really kind of changed my life. Before coming into SSP, I thought I wanted to be in the STEM space, but after SSP, I knew I wanted to be in the STEM space. And I also really found my people at SSP. And so, as such, this is maybe the most stressful audience I could be giving this talk to because the stakes are high because I care. So even though this presentation is also a little bit more philosophical than perhaps some of the other Curiosity Lab presentations are, give me some grace, please. I have not given this talk in a STEM dominated space. I've given it at like Facebook and Motorola, but those are very different audiences. So bear with me. Okay, let me share my screen. All right, and welcome to Guppies in the Boardroom. Okay. So I talked a little bit about working in the Natterson-Horowitz Medical Phylogenomics Lab. Just for a little bit more context, um, this is the book that got me into STEM and comparative biology and just biology in general. And it's a New York Times bestseller. And the woman who wrote it, Dr. Natterson-Horowitz, became my science Taylor Swift effectively after reading this book at age 15. She has presented Nobel Prizes. She's a Harvard and UCLA professor. Basically, I wanted to be this woman. And so when I found out that she worked at the LA Zoo, which was 8 minutes away from my house, I knew I had to reach out to her. And so 15-year-old me wrote a very, very nervous, cold email. And miraculously, she responded and agreed to meet with me. She had 15 minutes available 3 months from then. And I was like, great, I'll take it. Really, because this was just like meeting my favorite celebrity, right? And the meeting went on for 2.5 hours. And afterwards, she said, Noah, I don't really do this, but do you want to come work in my lab? Obviously, the answer was yes. And now we're here. So just before we get into the more specific stuff, just kind of defining what comparative biology is, because it's kind of a very specific niche. The idea is to take from pre-existing species and their adaptations and applying it to human health. And that can kind of be understood in this specific example. So take, for example, the condition atherosclerosis. That's the leading cause of death in humans because it's just high blood pressure, essentially. And the principle of comparative biology is to look at, for example, the giraffe, which has immensely high blood pressure, much higher than ours, but doesn't suffer from this condition. Why does it have high blood pressure? It has to push the blood all the way up the neck and back down. And because of that, the muscles and the arteries are super, super thick, and that raises the blood pressure, but they don't suffer from this condition. And so the principle of comparative biology would be to look at what other adaptations the giraffe heart has developed and then apply that to human cardiology, and you end up saving a lot of lives that way. So that's kind of the gist. In order to do that, though, we have to denounce this very long-standing kind of human idea that we're exceptional, that because we're like the dominant species on the planet, we did something right, we know everything, we can figure it out. And the way I think of it is like if you consider all of life on Earth as a flight from NYC to LA, humans come in about 500 feet before we land in LA. So we actually don't know everything. We know next to nothing. And nature has been doing this for much longer than we have. And nature has created a lot of adaptations to problems that we're dealing with currently. And so if we kind of take a step back and look around, you end up actually realizing that a lot of the solutions that we're trying to innovate ourselves already exist. Now we're getting into the more specific stuff. So we'll do a little thought exercise. Let's imagine a guppy tank, okay, with multiple males and a singular female. The female is the one that's kind of dull in colors, okay? This is what it looks like at first. After 6 weeks, this is what it looks like. No, the males did not eat the female. Don't worry, she's fine. She just masculinized, which means that she changed her colors to mimic males. She grew to mimic males, and she started behaving in a traditionally masculine way. Why is that, and what does that really mean? She's doing this because she wants to fly effectively under the radar of males to avoid, this is a kind of human term, but sexual harassment or unwanted sexual advances. This sounds very feeling-y for a fish, but the evolutionary explanation is actually very sound. An onslaught of unwanted sexual advances actually stops the female from carrying out important vital functions for her own survival. So being able to find food, hiding from predators, accessing other sort of resources. These are all things that she can't do when she's constantly being encumbered by a plethora of males. And so by masculinizing, she preserves herself and then the species can actually reproduce. So when I first saw this kind of phenomenon on a footnote of some other paper about male guppies, I immediately ran to Dr. Natterson-Horowitz and I was like, we need to do something with this. Like, this is really interesting. What are the human implications of this? Like, this is so cool. And she goes, no, this is really cute. But you need at least 20 species in which this occurs in order for it to be some sort of evolutionarily significant conclusion that can be drawn. And so I did a lot of reading, and we found 51 species in which masculinization in order to avoid sexual harassment has been identified. Masculinization occurs for a plethora of reasons. Like if you look at hyenas, for example, females will masculinize, but it's really more to assert dominance over males. And that, like, there's a lot of instances in which males will also mimic females in order to fly under males. Either way, this is a specific case, and we found 51 species, although this is not comprehensive. And why this is important, this phylogeny that I have on the screen, is because you can see that there's a phylogenetically diverse array of species here. So you have a lot of lineages. So you have invertebrates, which is all of the bugs. You have teleost fish, which would be the guppies that we were talking about. We have mammals, birds, reptiles. And what this means is that this evolutionary mechanism has existed either for a really long time, since we all had a common ancestor, or it developed separately amongst different lineages, which means that it was necessary despite all of the different selective pressures that each of these species undergoes. So this is where we get to the kind of the big term, the buzzword of the presentation, which is andromorphism. And that's just when a female organism has male physical characteristics or mimics males. And it's a potential adaptive strategy in response to unwanted sexual advances from males. And this comes in kind of different subsets. So you have behavioral andromorphism, which is acting more male, which we'll get into examples and you'll see what that looks like. Phenotypic, which is, the sort of visual male colors, whatever, growing in size. And then we have pheromonal, which is just either putting out male cues or suppressing female cues in order to go undetected. So now we'll get into some of the examples. This is the damselfly. The way damselflies anthropomorphize is both phenotypic and behavioral. So they will mimic male flight patterns in order to avoid detection, and they'll also have male colors. And this is actually effective to the point where even when most females look like males, males still don't catch on. And again, this is life-saving for the females because it allows them to access better spots on the pond, which is where, they live and also where all of their food is. It allows them to eat without being predated on, and it also allows them to avoid predators, even though they're brighter colored. You also have the hummingbird, which also displays male flight patterns in young females, and also the colorations of males in young females, females that can't reproduce yet, which means that not only can they not actually have offspring, but male harassment is actually detrimental to their survival, and often leaves them injured and unable to access resources. You have the fossa. This one is one of my favorites, mainly because people always think that I made it up and they say that it looks like a poorly drawn cat. I really like it though. For them, it's especially important because they're completely solitary. And so they live in very sparsed out environments. And so for a female to be constantly encumbered with male unwanted attention is lethal. But what they'll do is they've adapted faux male genitalia. So males will look at them and they'll be like, oh, I can't reproduce with this, and they'll leave them alone. And they also behave, especially young females, in traditionally male kind of behaviors and territory marking. We also have the brown periwinkle snail, and this is kind of the pheromonal one that I was talking about. So what they'll do is they'll suppress their female mucus cues when they're uninterested in attention from males. This is my favorite because I'm a big reptile nerd, and also because they kind of flip-flop back and forth. And so what the anole will do is males are typically green, females are brown, and what females will do is when they're like gravid, which means that they've already had their eggs fertilized, they just haven't laid them yet, they will morph into the green colors. And that way they go undetected by males until they've laid the eggs, and then they're ready and willing to receive that sort of interaction again, and then they will morph back to brown. There's also highly conserved genes involved in this whole situation, which I think is super cool. So 11B-HSD is conserved in humans and Danny-Oreiro steroid hormone biosynthesis pathways. Why is this interesting? Steroid hormone biosynthesis pathways are one of the integral pathways in masculinization in both humans and in teleost fish. And so though this is not an end-all be-all definite 100% link, it shows some central pathways underlying masculinization in other species and humans are significantly conserved, which goes back to what I was talking about phylogenetically earlier. This means that this has been a strategy that was developed way back when we had common ancestors and has not been selected out of either lineages, despite the different selective pressures that we go through, because, I mean, we don't live the same lives as fish. This is the slide that no one at Facebook ever asks me any questions about. I assume it might be a little bit different here. So if anybody has questions, let me know. But this is just kind of a little walkthrough of the methods and how we got here. We talked about the phylogenetic reconstruction a little bit. We had the fancy diagram, right, where we kind of scoped the literature, found where this phenomenon occurs. And then just now I was talking about the gene, right? So we selected 11B-HSD as a pathway of interest in masculinization in fish. And then afterwards, we consulted the Kyoto Encyclopedia of Genes and Genomes on 11B-HSD. And one of the pathways which showed up involvement in was the steroid hormone biosynthesis pathways, which immediately sparked my interest because I know that this is involved in human masculinization. Then we looked at the human steroid hormone biosynthesis pathway, but highlighted in green are the genes and enzymes that are shared with Danny-Oreiro or the zebrafish. And then within the steroid hormone biosynthesis pathway, we also highlighted the 11B-HSD in red. This is still the human pathway, by the way. And then finally, you have 11B-HSD highlighted in red from the human pathway and also highlighted in green in the species comparison pathway. So once again, you can see that it's conserved in both species and that it plays a significant role in the pathway that aids in masculinization, both in the fish and in the humans. So it's not a conclusive link, but it does, but it does implicate that it needs to be further studied and that there is a highly conserved genetic component to masculinization across species. Now we're getting to the more philosophical part, so bear with me here. We're getting to the boardroom, and I picked the boardroom because there are studies about it, but this happens in essentially any sort of male-dominated space, where what we see is behavioral andromorphism when you have the minimal number of women required to make your board look diverse or look good, or tokenism. And what happens is women will andromorphize to fit in. That means they will dress more masculine, they'll actually lower their octave of their speaking voice, and they will act more manly or macho in order to fit into the kind of boys club atmosphere. And it's actually been shown in this boardroom studies that the companies actually miss out on the benefits that women bring to the table when there's only one, because she's trying to conform to the male environment. But when you have at least 3 women, you actually get what women bring to the table. And they can be more authentic and successful and actually add the real diversity of thought and deliver value to the spaces that they inhabit. What does this look like? It's more collaborative spaces. You know, people feel more comfortable asking questions. They're more curious. You have more attentiveness. People are more open to listening, and they're conforming less. And this is all because you have a space where women can be in their femininity, and be authentic to themselves, as opposed to feeling like they have to conform to the male majority. And this increases the output of the corporation or the company or the space that they're occupying. So this is my favorite part because I'm very action oriented, right? The big takeaway for me here is that we are not guppies, right? The female guppy has to andromorphize because she's alone in her tank and she can't add more females, right? She can't balance out the ratio to take the strain off of herself. Humans are not like that, right? And when we talk about surviving, what we want is to actually thrive here, right? So we can pull more seats for more women. We can make these spaces more equitable, and reduce the effects of behavioral andromorphism in the workplace. What does this mean? It means that feminism is no longer solely an altruistic movement, right? It's not all about paying it forward and the sisterhood, though those are all beautiful things. It's about women wanting to promote other women for their own benefit and for the benefit of everyone sitting there. We're all benefiting when women can actually be women as they choose to be in the workplace, as opposed to us all trying to be this one thing that actually stifles the communal growth in general. So this isn't just going to help women be more successful, it helps everyone be more successful. That's kind of the spiel. If anybody has any questions, I'm also happy to talk about my current projects that I've been working on in the medical phylogenomics lab. Uh, Thank you so much for your time.
Thank you so much, Noah. That was amazing, and I learned tons, and you can tell that there's so much more to learn too. Um, Claire has some questions. Claire, do you want to unmute yourself?
Yeah. Hi Noah, this was awesome. I really enjoyed this. I have a bunch of questions, so I don't need to monopolize if there's other people, but I can use maybe start by going back to the phylogeny where you had all 51
Yeah,
species.
let me go back.
I guess my first question was, I know you mentioned there's like hyenas and others where the sort of observed masculinization is for a different like reason, quote unquote. Does this include like all of those? I mean, that you discovered?
So this is solely the ones that I found that are doing it to avoid unwanted sexual advances.
Mm-hmm.
Because masculinization and also the reverse feminization has been found in a lot of different species, right? So you have hyenas and you
Mm-hmm.
also have the jacana birds,
Mm-hmm.
are the two examples that come to mind. But it's not so much that females are doing this to avoid males, it's that females have higher levels of testosterone and traditionally male attributes, and they are actually dominant, and it's more of a matriarchy situation.
Sure.
So it's just kind of a different scenario.
Yeah, that makes sense. I mean, is there significance to this particular common ancestor of the bilateria?
This is what, once you input it into Philo-T, which was the software that I used to
Hmm.
track it specifically,
Hmm.
that's the common ancestor that it found,
Hmm.
um, and kind of calculates using the software.
Hmm.
But specifically, we don't have, unfortunately, any documentation of the specific common ancestor or what it was like
Sure.
just because it was so long ago.
No, that makes sense. Were you able to find if all the descendants of bilateria have this same behavior?
This is not comprehensive.
Sure.
So if I go and I look into every single one,
Hmm.
might find it, the likelihood is that you won't, just
Hmm.
because this is such a broad range
Sure.
of descendants, right?
Hmm.
You have a lot more mammals under this umbrella than are represented here, and that's
Hmm.
just generally mammal lineages are less plastic,
Hmm.
than teleost fish, right, or invertebrates in general.
Hmm.
So you're going to see less morphism, other than kind of behavioral in general.
Sure. But it looks like, I mean, even down to Dropsophilia, this behavior is observed.
Yeah.
Yeah, that's really cool. And then maybe can you also just explain what is a conserved gene? I think that might have been on just a couple slides after this.
Yeah. So conserved genes just means that it's a gene that hasn't been weeded out,
Mm,
right, evolutionarily. So for some reason, natural selection did not select it out, which means it was important for something,
mhm.
um, which is significant because if it didn't matter, we wouldn't have it anymore.
Yeah.
So then why do all of these species have it?
Mhm,
And this is kind of what we're talking about is like they have it because it's an effective survival strategy
mhm.
um, to avoid unwanted advances. Yeah.
I'm sure this is maybe a really research intensive question to answer, but did you find any species using your phylogenies tools that maybe have the gene, but you don't observe that expression of masculinization?
I mean, here actually, the one that I did find the gene in is actually not on here. So if you have Daniel Rario, for example,
Mhm,
is one that has the gene. That was the case kind of example that I showed with 11B HSD, but it's actually not on the phylogeny. So that would require probably another
I'm
whole
sure.
study onto why that's not expressed. Whereas, like in the guppy, for example, which is another teleost fish, but a different one, this is observed.
Mhm. Very cool. I have some questions about your methodology slides, but I'll open the floor to somebody else if they have questions, and then we can come back to that. Thanks,
Sounds-
Noah.
Yeah, of course. Thank you so much.
Claire, I think you're back up if you want for methodology.
Okay, works for me. Can you go to that methodology
Yeah,
slide?
here we go.
Yeah, is it just, can you zoom in or is this just
I
my?
can. It's a little bit blurry just because this was taken directly from the poster on which I presented this
Sure, that
on.
makes sense. I mean,
Um,
do you have is this from a specific publication that maybe you could link to or something? Because this is really interesting.
so this specifically, I can also share the poster
Okay.
with all of this stuff on the network,
Mhm.
just because this is stuff that I did using
Hmm.
KEGG, which is publicly available.
Mhm.
Um,
Yeah, I know,
but
I'd
yeah,
be interested. Yeah.
so wait, sorry, what was the question?
I
Was
think the
it
first question was maybe what you answered was where specifically this came from. So was this with your research with, I forget her name, your
Dr.
supervisor?
Natterson-Horowitz? Yeah.
Mhm. I mean, and so this is so her lab does this kind of phylogenetics work?
We've recently moved on to more kind of computational of a focus. So most recently, I've been looking into a lot of naked mole rats and the way that they process calcium without D3 because they're constantly underground. And the question is sort of why don't they develop osteoporosis as they age, which is something that a lot of humans struggle with. And
Mhm.
so the idea is they must have some sort of adaptation that prevents this because they literally can't process calcium or like in the way that we would think that they can.
Mhm.
So that's kind of something we've been working on recently. Another project that we finished and presented on in 2024 was sort of the evolutionary origins of genetically heritable diseases, which is the idea that we have genes that make us susceptible to disease because they provide us with some biological benefit or value.
Mhm.
Otherwise, we wouldn't have them anymore.
Right,
So that kind of goes back to the conserved gene idea.
right.
And I'm specifically looking into what's the evolutionary benefit of atrial fibrillation, which is like a pretty common heart arrhythmia.
Mhm.
And so calculating the overlap between the disease genes and also their biological benefit, especially now with the rise of gene editing, where there's like this growing consensus, we can just cut disease genes out. Like, that's really cool. That's really great. No, we can't.
I know
uh,
I'm
Noah,
trying.
could you share where this was presented?
Mm,
oh, yeah. I said it earlier, but I can say it again. It was presented at the International Society for Evolution Medicine and Public Health.
very cool.
Um, this one was in Irvine. The project I just talked about was in Durham.
Mhm.
So we've been all over.
Have you ever read, there's just a book, it's a pop science book, but it's called Survival of the Sickest. It's the same concept that hereditary diseases that have remained among the gene pool must have had some selection pressure in favor of them. Although I think it focuses more on more recent human ancestry. I don't know how rigorous it is, but I think it's a pop
That
science.
sounds really cool.
Yeah,
Yeah.
it's a pop science thing. I know he did a lot with diabetes and I think hemophilia as well, but some of those others that really appeared in some of the European royal lineages.
Hmm.
Um, yeah, very cool.
Yeah.
Thank you, Noah. I mean, I have more, I think this is really interesting. And, like Alan, I was in astro, so I don't have a biology background.
Oh, very cool.
This is interesting to me, so I can keep.
I really admire all of the astro stuff. Like I heard from some of my friends who were in some of the astro programs, like the sleep schedule is crazy because you're out and you're observing stuff, and I'm like, respect. So that's a whole separate thing. But
Well, maybe can I ask, I mean, so you're headed off into Stanford. I mean, what do you have a lab in mind that you'll work with or a direction that you're specifically interested in?
right now I'm into, I guess, bioinformatics
Mhm,
as used for biomedical kind of
mhm,
engineering, right? So ideally, where I would essentially go is kind of taking this stuff
mhm,
and then looking for the specific genes and like species of interest
mhm,
that we hypothesize would have adaptations to that, and then eventually being able to target those genes and kind of create primers,
mhm,
right? So that you'd be able to eventually CRISPR those genes into either like mice for experiments and see if they develop higher resistance to these things. And then eventually the end goal is like humans, right? And then you end up hopefully developing new treatments to
sure.
diseases that we haven't really, I guess, been able to tackle completely.
Yeah, fascinating stuff. Great.
This has been fascinating. Thank you so much. Um, thank you, Noah, for your time. I'm excited to post this as well on the SSP network as well as the YouTube, so you'll be able to access this in the future. Um, and then if you're ever interested or if this sparks some interest, then please email me at jkuhn@ssp.org. And then um, uh, we're always open to other presenters and things in the future. So thank you, Noah, and have a great rest of your day.